Today we have invited Martin Ooms from the Belgian Nuclear Research Centre to talk about the research into radiopharmaceuticals for cancer treatment. So this seminar is being live streamed on YouTube and it is recorded as well. So if anyone has a problem with that, then you can just let us know. If anyone has any questions at any point, then we'll have a Q&A afterwards. Or if you want to ask Martin during the talk, you can put your hand up and then I can relay the question. Okay, is everything okay for you to start, Martin?
I think I'm ready to start. If you guys hear me and you see my slides, then I'm ready to go. So yeah, I tend to, if I start rambling, I mean, if you have questions, feel free to interrupt. In the meanwhile, I'll keep talking until I hear otherwise. Good. So yeah, I would like to take this seminar to have a bit of an overview of radiopharmaceuticals, explain a little bit what they are, how they can be used, and how I want to give an example of like how a radiopharmaceutical development process can start or can be monitored. So yeah, the outline will, as I mentioned already, so we'll talk about what they are, how they can be used, and the radiopharmaceutical development, specifically what we have been doing here at SCK in the past few years. So let's start with the beginning. The basics of radiopharmaceuticals are, as the name suggests, radioactive drugs. So, and they're composed of two major components. So first of all, you have a targeting vector, which directs your radioactivity to a certain site in the body. And then on the other hand, you have a radionuclide. So that is the payload and actually delivers whatever property or physical property or damage you want to do to your target. And then you have a linker that basically links the two together. So I'll go a bit in overview. So of course, the easiest way to understand this is to, like, you're carrying is a bit of a car with a GPS in it. It knows the way and you can, it drives you to the tumor site, for example. You have the radionuclide that is a payload, and if you want the radionuclide to hitchhike together with the target vector, we need some sort of stroller or something we can connect to the car to get it to the parking lot. So that's a bit a very simplistic way of explaining these things. Now, if we look at it, there's a few prerequisites or important things to know. So first of all, the target itself, of course, you want it, your target to be expressed very highly in the target tissue and very low in healthy tissue. So if you want to, for example, have target a tumor cell, you of course need to pick a target, a molecular target that has very high expression in the tumor, because otherwise you will not distinguish between the tumor and your healthy tissues. These targets can be anything, can be receptors, it can be transporters, it can be intracellular enzymes. So anything that basically distinguishes your cancer cell or your target cell from your healthy cell can be used as a target. Now, if we go to these carriers, of course, they need to bind specifically to the target, but also for these carriers, we can have all kinds of options. So for imaging, often small molecules are used. So they are quite small and they can be basically mimicked towards the product or a physiological compound. For example, you have here the fluorine-18 glucose derivative that's often used. On the other hand, there's also a lot of focus moving towards more biomolecules. So peptides have been used a lot, antibodies and all kinds of antibody fragments, nanobodies have been used. As well as like nanoparticles, liposomes, all these things. Everything that has a certain affinity towards a tumor can basically be used as a carrier. Not all of these things are as easy to use, but at least they're possible and they have been used. Now, of course, we'll come to the important part, and that's the radionuclide. So that basically is the part of the radiopharmaceutical that gives us the properties that we want. And for radiopharmaceuticals, we can distinguish basically two major radionuclides that are used. So on one end, we could decide to emit or to connect a gamma or a positron emitter to that thing. So basically, we can connect, conjugate target a technetium-99 or fluorine-18, which are positron emitter or gamma emitter, and then we can use that for SPECT and or PET imaging, depending on the isotope. So we can really visualize the radioactivity from outside. We can also choose to attach a nuclear bomb to it with alpha emitters or beta minus emitters, and then we call that target radionuclide therapy. We can actually use it, the therapeutic potential of these radionuclides in a directed way in the radiopharmaceuticals. Now, of course, it is important. You have the radionuclide, you have the vector, but of course, you need to make sure that these things are also stay connected. So there needs to be a linking between the radionuclide and the vector. And that type of linking depends a little bit on the radionuclides you're using. So for fluorine-18, carbon-11, these can easily be covalently bound to carrier molecules and easily can be put in. For example, carbon-11 is often just conjugated with a methylation into a precursor molecule, and to form then basically exactly the same molecule as the molecule you want to mimic, but then a radioactive version of it. However, if we use radiometals, and that's often used in therapeutics, we actually need to use a chelator where we try to build a cage around to hold the radionuclide that has a very strong affinity towards the radionuclide, binds it, and also very importantly keeps it bound, because at the moment your radionuclide is lost from the chelator, it will also not follow the carrier anymore, because that's a very important thing to pay attention to. So in a very nutshell, radiopharmaceuticals, to quickly sum up again, so we have the targeting vector, which is basically determining where your radiopharmaceutical is going. You have the radionuclide that determines what effect your radiopharmaceutical will have. You can have or imaging, or you can have therapeutical radionuclides. And then you, of course, need a way to conjugate the two by a chelator, or you can have direct radiolabeling depending on the radionuclide. So now, to give you a bit more an overview of how these things can be used, because it's all very nice in theory, but there are some real applications that are coming from clinic already for quite a while that can prove the use of radiopharmaceuticals. And for that, I want to give an overview of two major applications in nuclear medicine, and that's in molecular imaging, basically using the imaging isotopes or the targeted radionuclide therapy using the therapeutical isotopes. So if we let's kick off with the molecular imaging. So what we do with molecular imaging is basically that is a process to visualize visualize biological processes in vivo. So what does it mean? You all know a CT or an RX where you get anatomical information. With molecular imaging, you will not get that. You will get info on a biological process. So basically what you will do is you will inject a radiopharmaceutical into a patient. It will automatically accumulate in the tissues that you are targeting. So in this example, if you use tumor tissue, you basically it will accumulate in the tumor. But the nice part about this is because you're looking at biological processes, that the metastases, who are basically most of the time biologically the same as the main tumor, they will also accumulate these radiopharmaceutical meaning that they can light up in an imaging. So if we then take a camera to visualize the gamma rays or the radioactive distribution in the patient, we can visualize the tumor uptake, and we can really see tumors that you cannot see on CT or conventional imaging techniques yet. So you can much better pick up things. So there are two types of cameras that were used. I already mentioned the PET and the SPECT. I will not go into the details about it, but the PET scanner is basically used for positron emission, where you make use of the two anti-parallel gamma rays that are emitted in the beta decay, while SPECT is another way where you try to use only the single gamma line emissions, where you try to filter it perpendicular to the camera. Now, as I mentioned already before, I showed it in the slides earlier. I think one of the key, or that has been along the workhorse of the nuclear medicine these days, is FDG. So it's basically a fluorine 18 derivative of glucose. So as you can see, it's actually the same molecule, but instead of hydroxyl group, they replaced it with a fluorine 18 group to get a radioactive version. The nice thing is it behaves exactly the same way as glucose, so it will also be taken up by cells that use a lot of glucose. So another advantage is in the fact that, so if glucose is taken up in the cells, so you will basically have hydrokinase phosphorylating it and get into the Krebs cycle and to basically to burn up the glucose. For glucose itself, this is a reversible process. So you can basically switch easily between the glucose and the glucose 6-phosphate. However, with FDG, that's not possible due to that good location of fluorine 18. Your enzyme cannot recognize it anymore. And basically what you have is, once FDG is in, it's a bit of a trap. It's a one-way street. And that means that your FDG is really, while glucose can migrate over the membrane a lot, of glucose, once it's entered, it's trapped inside the cell. And that means that you can really have radioactivity accumulate in cells that are highly consuming glucose. What are these cells? Of course, you immediately think of cancer cells. So cancer cells really, they grow a lot, so they burn a lot of sugar to keep up with their metabolism. And therefore we use, it's often used in oncology. So you can see, these dark spots where you have some hot pockets of tumors or metastases that show accumulation of radioactivity. Of course, other organs that use a lot of glucose are the brain. So the brain also takes up a lot of glucose in normal, so because it's only using glucose. However, for example, with certain diseases, Alzheimer's disease, for example, or addiction, there's really a shift in brain activity, which can also result in changes in glucose uptake. For example, in Alzheimer's disease, with the normal brain, you see the radioactivity distribution all over the brain, or the cortex at least. However, with Alzheimer's disease, there is a specific pattern of degradation where the glucose uptake is actually decreased before onset of the clinical Alzheimer. So even earlier, you can already see changes in the brain happening before we can physically see something, or we can do it through other imaging techniques, or even sometimes clinically can be detected. It's also often used to take a differential diagnosis. So different types of dementia have different types of patterns that close down in the brains first. Also in cocaine addicts or drug addicts, there's a lot of changes in the brain metabolism that can be mapped through FDG. And then finally, another type of cell that uses a lot of glucose are the immune cells. And so there also can be used to visualize inflammatory effects. So here you see an image of so an artery inflammation that can be clearly seen on a PET scan as well. Of course, if you can use it for diagnosis, you can also use it for disease follow-up. And that's the nice thing about FDG. So if you look at this patient, this is a pre-treatment patient, which has all these black spots. You don't need to be a medical doctor to see that this is not really a very good scan. So what you see is all these hot spots, or a lot of these hot spots are metastases. What you can do is then you can do an imaging study before you start your treatment, and then you could do your treatment and then basically follow up the same patient in using scans. So basically, if you look already at the difference between that cancer patient before treatment and 14 days after treatment, you already see a lot of difference in glucose uptake. That doesn't mean that the cells are not there anymore. They're still there. So on anatomical imaging, you will not see a difference yet, but you already see the shift in metabolism. So your cells are struggling, they use less glucose, so you have a therapeutic effect. So that means that you can basically play a lot closer. You don't have to wait for your tumors to actually shrink to already have an idea on the therapeutic efficiency of your treatment. And that can be often used to make sure that you have these in between thresholds to check, okay, do we continue with this chemotherapy, for example, or do we stop and change? And that can be done a lot faster than in other images. So, and then you can keep following up this patient and you can clearly see changes and the FDG activity decreasing over time. So that's a bit how imaging is worked. There are also going to be a lot of research because you can visualize these biological processes. I'm not going to go into too much detail in that because then we're talking for yet another hour. So I'll move on to now how radiopharmaceuticals are used in target radionuclide therapy. So if you look at it, the cancer treatments there's quite a few options for cancer patients these days. Of course, the first line is always surgery. If you have a localized tumor, you will want to just dissect the tumor out and basically cure the patient. However, the moment there are some risk of potentially metastases, often they combine surgery with a second line of treatment, and that's chemotherapy and radiotherapy, so external beam radiotherapy. Lately, there have been quite some new therapies that have been added to the portfolio of cancer treatments, really trying to let the immune system fight the cancer cells themselves, or really train the immune. So immunotherapy or personalized medicine, trying to figure out what exactly is going on in that specific tumor, in that specific patient, and then try to figure out a way how to tackle it and how to make the life of that tumor difficult. So, and somewhere in between, there's also target radionuclide therapy that basically combines a little bit the personalized medicine with radiotherapy. So how does it work? So compared to, it's often compared to conventional radiotherapy, where they apply externally a very high radiation dose. So they will try to target your X-rays or your gamma rays as closely as possible and as precisely as possible towards a tumor. So they try to focus on the tumor, and they try to irradiate from different angles, but always the tissue before and after your tumor will also obtain quite some damage. You will have a lot of burns and skin burns, etc., from irradiation. So that's one of the key complications here, or difficulty. Additionally, radiotherapy or external beam radiotherapy works quite well in an early phase of the disease. So if you have like the main tumor and maybe one or two metastases, you can still do that. But the moment the disease is highly spread and you have a lot of metastases, you cannot irradiate the entire body. And that's where target radionuclide therapy can actually have its strength. So as I mentioned before, because we use biological processes to target the cells, we basically can inject radiopharmaceuticals that really target cells and specifically accumulate in the cancer cells and basically irradiate the cancer from within. If we can then make sure that our radiopharmaceutical has the right pharmacokinetic profile, we can actually try to minimize the residence time in the body and try to maximize the tumor accumulation. And that way we have actually rather limited damage to healthy tissue, of course, provided that we get a good pharmacokinetic profile. So one of the major breakthroughs lately in prostate cancer, or in target radionuclide therapy, has been made on prostate cancer. So prostate cancer is the most abundant cancer in the male population, and it's actually characterized by very high survival rates, as soon provided that you know it in time. If you find it in localized stage, the odds of you surviving are quite high. Also, if it's still not really spread too far, it is. But the moment it starts really spreading and you get distant prostate cancer, your survival odds drastically drop. So, and what is good for this tumor, or these tumors actually also have a high expression of a prostate-specific membrane antigen, what we call PSMA, which is basically a very good basis. It's not really expressed on other tissue, so it's a really good basis for targeted radionuclide therapy. So, and what they did is they developed a compound, the PSMA 617. The numbers don't really matter, but it is a compound that binds specifically to the PSMA receptor. As you can also see, there is a chelator here, so we can basically radio label it with some metals. It has been radio labeled with lutetium-177, but the more dramatic results have been shown with actinium-225, which is an alpha emitter and which has a very high potential for treatment. So, and to showcase a little bit, there are a lot of these images, but to showcase a little bit of the potential of actinium and actinium PSMA targeted radionuclide therapy, I want to show you some published results that were obtained in very late stage prostate cancer patients. So these were patients that you can judge by the images already, were full of metastases, bone metastases everywhere. So these were patients that were really end stage. They had a few treatments already without any success. So what they did in the end, they tried to say, okay, let's try to treat it with actinium PSMA. What they then saw is quite impressive. So after a few cycles, you basically not only see that the metastases are gone, but you can also see that the PSA, which is basically the tumor marker that's used to follow up patients in blood, is drastically dropped, and actually is dropped to the levels of a healthy patient now. So it's really impressive, these data, and certainly judging if you have these patients that had maybe 1 or 2 months to live to be kind of cancer-free after a year, it's really impressive. And that's also set out to believe that there's a lot of potential in there. Of course, there's also a bit of a toxicity involved in this actinium therapy. So that's something we're trying to solve now, but it can show what great potential targeted radionuclide therapy can have, certainly in metastases and metastasized cancer, so late stage cancer. So we've talked a lot about radiopharmaceuticals and what they are, and I would like to now then finally go over what it takes to develop these kind of radiopharmaceutical. And as an example, I would like to give a bit of an overview and a bit of the story that we've been working on here at SEK to give you, to understand what this radiopharmaceutical process development process looks like. So within the NURA program, that's the name for our medical nuclear medicine research, we're actually focusing on therapeutical radiopharmaceuticals. So we're trying to bring know-how to all fields of therapeutical radiopharmaceuticals, and that includes, of course, we need the isotope. We have a nuclear reactor on site, which is convenient if you want to produce a lot of isotopes. But also looking at trying to find new radio labeling techniques, trying to have new chelators to conjugate a radioisotope to the carrier of your choice. We then, of course, also need to work on preclinical evaluation. Once we have that radiopharmaceutical, we want to know, does it work? Does it do what we want it to do? Does it accumulate does it go where we want to go? And then we also have a few groups working on the radiobiology. So what really happens in biologically speaking with these cancer cells after exposure to these radiopharmaceuticals, but also what happens in healthy tissue and how can we protect healthy tissue while targeting the cancer tissue still. And then finally, of course, a lot of all the therapeutic indexes depend on your dosimetry. So you want to find the optimal dose or the optimal amount of radioactivity you need to inject in each patient to find the optimal therapeutical index. So if we start, I want to start a story, of course, from the isotope, because that is in some way the most important part of the radiopharmaceutical. And that is SEK. As I mentioned already, we're mainly focusing a lot on therapeutical radionuclides. So we have a few isotopes in our portfolio: lutetium-177, which is often used. We have samarium, rhenium, and also we're collaborating with Pantera, which is a joint consortium between SEK and IBA on really upscaling and getting high amounts of actinium for clinical patients. However, what I would like to talk to you about is a bit of the Terbium-161 story, which is a bit what we consider now our flagship isotope, and that we try to produce in large amounts. So our terbium, Terbium-161 is a bit of a unique radionuclide in a way that within the terbium series, so there's a lot of brother sister isotopes. I don't know whether that's male or female, but so brother or sister isotopes, which actually have the same chemical element. They're all terbium isotopes. However, they basically span the entire spectrum of nuclear medicine. So you have the Terbium-161, which is a beta minus emitter, which can be used for beta therapy. But on the other side, you also have the Terbium-149, which is an alpha emitter and can be used for alpha therapy. And then on the other side of the spectrum, you also have the Terbium-152 and the 155, which are positron and gamma emitters, so they can be used to PET and SPECT imaging. So with the same chemical, exactly the same chemical, the same terbium in your chelator, you can actually, by changing Terbium-161 to 149, 155, you will not change the properties of your radiopharmaceutical, biologically speaking, but you will be able to plug and play whatever you want to do. On top of that, terbium itself, natural terbium, also has some fluorescence properties that could be used if wanted, but then often the concentrations we use are not high enough. But this is added for completeness. So Terbium-161 is so chemically, I mentioned already maybe that currently all therapy in clinic focus on lutetium-177. So Terbium-161 is also a lanthanide. It's quite chemically comparable to lutetium-177. It has similar physical properties. So the half-life is approximately the same. The beta energy is not that far off. However, there is a much higher emission of Auger electrons. That actually, these are high linear energy transfer isotopes, particles that give up a lot of dose in a short distance. So in that perspective, terbium can be a very superior potential, superior compared to lutetium. And the first indications in studies that have been published, mainly by the groups of PSI in Switzerland, they have been showing that when they do a head-to-head comparison with lutetium, terbium with the same dose gets much better results than lutetium. So there's a lot of potential. However, there's a big limiting factor, and that is the supply. So far, when we started, PSI was supplying a little bit for their own use, but not really a lot of supply. So if we wanted to work with terbium, we had to figure out a way to produce it ourselves. And therefore, it comes in handy that we have our nuclear reactor on site. So basically, there's a quite easy way, straightforward way to produce Terbium-161. And that is, if you have a highly enriched gadolinium-160, you can irradiate that with neutrons, put it in a high flux reactor, and you basically generate gadolinium-161, and that gadolinium-161 automatically decays into Terbium-161. And then you have two isotopes that you can separate. So if you look at the entire process of such a production, so of course it all starts with the gadolinium target material. We need to be able to we had to figure out a way how to get the gadolinium inside the BR2 reactor. So we need to get it into the ampules. We looked at what kind of material do we irradiate, nitrate, oxides, these kind of things we try to evaluate. To then come up with an irradiation protocol. So it would be irradiated in the BR2 to get then a mixture of gadolinium, like a lot of gadolinium-160, and chemically speaking, not a lot of terbium-161. So then we had to develop a radiochemical separation process that basically allowed us to extract these very few atoms of terbium 161 from a huge mass of gadolinium. So it's a ion exchange chromatography system with big columns to get that separation done. So, and then to get to the terbium that we can use for radio labeling. However, I don't know if you ever worked with highly enriched materials, but they're extremely expensive. So of course, we cannot just throw them away after one irradiation. So there's still a ton of gadolinium. If you have a 1 milligram target, it will basically still be 1 milligram of gadolinium coming out. So we of course need to figure out a way how to can basically not only extract the terbium, but also recover the gadolinium from that system to bring that back into our target material, to have a closed circle economy. That's the buzzword these days they use. So of course, as I mentioned, PSI has been working on terbium, and the research was mainly limited to well-known carriers. So dotatate, the PSMA that I mentioned before, there had been some folate, and they were all quite heat resistant. So they were radio labeled 90 degrees. They basically boil it up for 15 minutes. However, that worked perfectly for these carriers, but the radiopharmaceutical field is more moving towards these biomolecules. So antibodies, nanobodies, or antibody fragments, which are not that heat resistant. So therefore, we had to figure out a way to radio label these radiopharmaceuticals without the harsh conditions. And that did not work with the radionuclide or the chelators that were available at that time. So therefore, we've set out to find a new chelator for terbium, not only one that chelates, but also chelates at mild conditions. So we tried a few chelators. Some of them are commercially available. This one, for example, the DOTA, is the one that has been reported previously. And basically, some others we synthesized ourselves. And what we see is we did some radio labeling tests where we tried to radio label with different molar activities at 40 degrees, which is a cutoff where most biomolecules still can live. But if you go a lot higher, some biomolecules might get in problem. And what you can clearly see immediately, you do not get quantitative yields with DOTA. If you use the DOTA chelator, as was we already taught from previous literature, you do not get these quantitative yields. Maybe to explain quantitative yields is more than 95% radiochemical yield. That means that if we add terbium to a radiopharmaceutical or to a precursor, that 95% of the terbium is automatically incorporated in that radiopharmaceutical. Now, why is that 95% important? Is because that's what we consider as stable and as okay in good enough purity to inject as such in the patient. So if we don't reach that 95%, we have to add a purification step where you might lose a lot of your radioactivity in filters or in cartridges. So that's not really what we want. So ideally, we want quantitative yield. So of course, but getting into the chelator is one thing, but we also need to make sure that it stayed in there. As I mentioned already before, it is important that that your radionuclide stays close to your chelator and therefore also close to your radiopharmaceutical. Otherwise, you will not have that effect where your radionuclide goes along with the isotope, and it goes its own way in the body. So therefore we did some stability studies. So first of all, we incubated our radiopharmaceutical in human serum. And there you already see that one of the chelators is struggling. So you have the DTPA, which is an heterocycle, acyclic radio carrier. Does not really, starts already to show some instability in human serum, basically meaning that you have high purity. However, if you incubate in serum for a while, you basically have some exchange from ions in the blood or in the serum to the chelator, so you'll have loss of leakage of your terbium out of the chelator. We did the same in PBS, where there was a bit higher stability, but of course there are not that many metals present in there. So yeah, judging by that DTPA, we already figured out that's also not really the chelator we want to work with. Now, of course, this is all in vitro setting, and it's all very artificial. You can incubate it with serum, but that does not mimic the radiopharmaceutical streaming through the bloodstream and getting there in different compartments. So what we wanted to do is we wanted to then conjugate it to a compound that actually then we can do a stability test. And therefore we conjugated it to human serum albumin. What is the advantage? Human serum albumin is basically something that stays migrating into the blood. So it does not go anywhere. It does not have any affinity towards something. So it circulates into the blood. So that means whatever you see in activity, if you see accumulation in activity in some organs, then you know that's actually coming from release of your terbium from your carrier, from your chelator, and not really from accumulation of a radiopharmaceutical. So if you look at it, and I want to specifically point out to the bone here. So if you look at, if we did this biodistribution, where we basically injected in mice, and then we sacrifice the animals after certain time points, dissect all the organs out, and dissect all the organs out and then count all the activity in each organ, we can clearly see an increased accumulation in the bone here with the DTPA, the compound which is not stable, while this is not the case for the DOTA-GA, for example, the other chelators. And why is this bone so important? That is actually because of this. We also try to inject the free terbium chloride. And if you notice here, if you inject terbium chloride, it will immediately go to the bone. So the moment terbium is released from its chelator, it will seek the bone and starts accumulating there. So what we see here in the previous slide is actually accumulation of free terbium after release from the chelator. So then to the final part, of course, we now have a chelator. So we did not really have a radiopharmaceutical yet. And therefore, I cannot really disclose the nanobody because of, we have some contracts that we cannot mention a lot about it. But then we started to working with nanobodies. So we decided that the DOTA-GA was a compound to go with. And what we did is we first needed to conjugate that to a nanobody. So what we used is basically these nanobodies were modified. So we had a C-terminal cysteine, which resulted in a free thiol group that easily reacts with a melamide moiety. So, and basically what you do is you bring these two together, and then you get your nanobody, that is your chelator that's conjugated to your nanobody. If you then do your radiolabeling, you can then add your terbium to it. You have the radiolabeling process that we evaluated earlier, 40 degrees. You can do radiolabeling, and you can clearly see here, this is a size exclusion chromatography, that your terbium is highly associated with the nanobody. There's always a little bit of terbium that did not react, but that was less than a few percent. So we were very happy with these radiolabeling results, showing that, we really could radiolabel our nanobody with terbium. Next step is to do some in vitro evaluation. So we need to show the specific binding, and we want to confirm that it binds specifically. So we added it to BXPC3 cells, which are cells which highly express the target of the nanobody. And we could clearly see that we had specific binding in these cells. And also, if we incubated these BXPC3 cells in the presence of radiopharmaceutical, we already saw some biological effects. We saw a decrease in viability, and also the colony formation was decreased. So we can really see a therapeutical effect. And then finally, we also did the same biodistribution where we injected the radiopharmaceutical in mice, and we can clearly see a high accumulation in the tumor, which is nice. Of course, you also see kidneys, as you also see with other biomolecules. So that could, in the end, provide troublesome for the dosimetry, but it's something we need to figure out. But also in the SPECT images, you can clearly see your tumor lighting up in the SPECT image, which is nice. So, and then finally, that's the very last 2 slides I want to look at. So what we're doing is we're working with nanobodies and conventional carriers, but we're also looking at more non-conventional carriers. And there I want to highlight the project of one of our PhDs, where we're trying to work with nanoparticles that are often used as MRI contrast agents and something like that. And so often, these nanoparticles are quite often used, and they have been used in radiopharmaceuticals as well, but often they then use a chelator that is then connected to a radiopharmaceutical, a chelator that's connected to some of the outer shell, and then we can conjugate terbium to it. What we now wanted to do is actually to, instead of having your chelator there, and you have the risk of losing your radionuclide from the chelator, we actually wanted to make the nanoparticle itself radioactive. So that would in theory have improved stability versus traditional chelators. It also, the nanoparticles itself, we can have them targeting and have targeting vectors on them, but the nanoparticle itself will also have an enhanced permeability and or improved targeting. And finally, the theranostic, so they can also be used as a theranostic. So you could combine it with an MRI to both use a therapy and follow up where your nanoparticle actually goes using MRI to test that. So it's an idea that we've been working on. We've been doing the first optimizations. We've been doing the radio labeling these days. I cannot really, the data are still preliminary, but we actually had quite good radio labeling yields now to get these nanoparticles. The reason why I'm saying is that it's just a bit, it's more than biomolecules only. It's more than carrier nanobodies and antibodies, but there are quite a few other things that we can do. So then I come to the end of my presentation. I hope I didn't speak too long, but I would like to thank you for your attention. And of course, I'm always happy to answer any questions or start any discussion to explain you more. So thank you for your attention.
Thanks very much for that very interesting presentation on the Neuro project, Martin. So just to kick the questions off, I had a question to start with. You briefly mentioned the dosimetry. I was wondering, how would you work out, when it comes to the stage of testing the radiopharmaceuticals on patients, how do you work out the dose for a specific patient given a specific cancer? Would you use like phantoms or?
Yeah, so currently the current practice is that this isn't done. They basically, you have the patient, and the standard dose is 200, well, I have to say the gigabecquerel these days, 8 gigabecquerel or 7.4 gigabecquerel. So 200 millicurie is the standard dose for lutetium. They give that to any patient. If you really have a big patient, they might give you a bit more. If you have a little patient, you might get a little less. But in general, it's very ballpark how it's done now. But that's a bit where we also want to get rid of. I didn't focus a lot on that in our presentation. But what we're trying to do here as well is to figure out, okay, first of all, you need to look at your distribution. You need to know where your activity goes, and that can be quite well extracted from a pre-treatment scan. So what they do in the clinical practice is first they inject for dotatate, for example, which is a therapeutical agent. Lutetium dotatate is used, and it's actually even accepted by the EMA, the medical agency, reimbursed in a lot of countries. So what they do is, before they actually inject the lutetium compound, they actually make a gallium compound of it that they can, gallium-68, they can image. They can already see patient selection. Okay, does this gallium dotatate, is that taken up in the tumor, yes or no? Because if the gallium dotatate is not taken up, the lutetium dotatate will also not be taken up, and that's basically a waste of resources to do that. Then you don't. So the moment if you have a negative gallium dotatate scan or gallium PSMA scan, you will not get a treatment because you already know you will not benefit from the treatment. But what we're trying to do is there's a lot more you can get from that scan. So you can look at distribution patterns, and what we want to try to understand a little better is how the activity that is in the kidneys, for example, relates to kidney damage. And that's something very hardcore basic research that still needs to be done, and that's where dosimetry and radiobiology come in. So to have an idea on how much activity is in there, then quantifying that activity, because from a scan, and that's where phantoms come in. So you will have to somehow mimic your body and try to, with a known activity, know what the kidney would look like and try to get some phantoms there. So that's one side. You need to quantify it, and then you need to look at the biological effect or the biological relation between that activity and transferring into actual biological dose.
I imagine there's a lot of spherical objects filled with water involved with the first.
Now it's well, the best phantoms start already mimicking some of the major organs, but they're quite expensive, these phantoms, and there are for our preclinical scans, we use an Eppendorf tube with a little bit of activity in a pool, like a Falcon tube with higher activity. That's our phantom these days. But yeah, it's something that really needs more attention, and the attention is coming. And the field, they start to realize that they cannot continue the way they've been doing. So the realization is there, but it takes a lot of time to really get, and also a lot of, and that's maybe the bad side of it. It's difficult to also get funding for it. A hospital gets reimbursed a certain amount for that treatment. The lutetium, they get a reimbursement amount. They cannot really spend a lot of money on top of that. That's often a problem.
Sorry, we have an alarm going off, which is weird. Okay, I think it was just the weekly fire alarm. Sorry about that. Okay, does anyone else have any questions for Martin?
One in the chat.
Oh, okay, thank you. So we've got a question in the chat. Just for the benefit of the room, what techniques have you been using for monitoring the purity of the compound to ensure that it's over 95%?
Yeah, so it's, we use two major ways, or it's an HPLC method. So that's, depends a bit on the compound you're radiolabeling. It's usually like a C18, C3 column, or size exclusion. For the nanobodies, you often use size exclusion chromatography, where you need, and then you can do a quality control of your sample. So you take a small sample, inject it on the HPLC. Another way that you can work with is with TLC, ITLC. So where you spot it on a TLC strip, let it run, and you basically separate. You find the methods where your free terbium migrates and your bound terbium does not migrate, and then you can basically visualize the activity on the TLC. You can cut it in two and quantify the top and the bottom, and that's how you can determine these yields. So it's chromatography techniques, ITLC, so thin layer chromatography, or HPLC.
Thank you.
Will we be able to say anything here?
Well, if you ask the question, I think
if you talk loud enough, I can hear it, but
Now,
can you hear Daniel okay? Would you like to try and then
sure.
I'll relay it otherwise?
So my question is also on the purity of the compound. I was just thinking that 95% seems like far too much of a convenient number. Is it something that was actually like a generic number set by a medical institution, or is that a number that has been advised by experimental results? And if it's the latter, what happens once you go lower than that 95? What are the adverse effects to the patient?
Yeah, so first of all, that number 95, it's been a number that's been passed through generations. But I think it's more of a, they can, yeah, it's also picked up in pharmacopoeia and these kind of things. So it's more of, I don't know, generally accepted limit or acceptable limit. What happens if you go to higher impurity rates? So the problem is that, so the impurities, certainly in chelator-based radiopharmaceuticals, you can have two impurities. So it's or the free radionuclide, or it is the bound one. And of course, if you have a lot of free nuclide, let's say in extreme cases, you have 50% of your radiopharmaceutical, you have 50% of free bound, or free radionuclide. So what happens if you then inject 1 gigabecquerel, you will only be able to control 500 megabecs of that. So half of your dose, you will not be able to control. It will just be the terbium going through, as we saw, to the bone, with all bone marrow toxicity and stuff. So the side effects depend a bit on what radionuclide you are working with. So for example, bismuth 213, that really goes to the kidney. If you're looking at, yeah, it really depends on the isotope itself, but the problem is that you do not get proper targeting anymore. And for some, yeah, at some point they accepted 5% of your radioactivity not going, maximum 5% of your radioactivity not going where you want it to be is still acceptable.
Thank you. Any other questions? I had one more. I might regret asking this because it might be too complicated, but I thought it was really interesting what you were saying about the glucose uptake in people with addictions and with Alzheimer's. Why does that vary in those cases?
Well, so that's, then we're talking about neurobiology of Alzheimer's disease, for example. But what happens, and I don't think they fully understand why, but there's different types of dementia. And if you look at clinically, they're also a bit different. So you have types of dementia where you lose speech first, that you still remember stuff, but it's more difficult to talk. You have where your short-term memory is affected, you have your long-term memory, and they're all different regions in the brain. How these things differ, I honestly don't know. I'm not that expert in the brain biology, but what you do see is that these also correspond to the regions, or the regions that are taught to control, for example, speech. They will drop out in FDG also first in speech impaired dementia patients. So an Alzheimer's disease has a specific, yeah, it also follows. So Alzheimer's disease is known by some amyloid plaques that basically get on the brain. So it's sort of scar tissue that gets, and it follows a certain pattern. I don't know why that is. I don't know if it's even know why that is. But basically you see the same pattern with FDG. So you also see that the glucose metabolism drops in these regions where these plaques are forming. So that these get less active, and that's also represented then in the clinical stage later. But often you already see your glucose levels drop a little bit. They're still active, these brain cells, but are already struggling. You see that they're struggling. While that might not be clinically relevant yet, you will not notice that patient. But there are some instances where they scan the patient and they actually said, like, yeah, you might get Alzheimer's disease for another study, yeah? But that they figured it out that way, or that they at least had some severe indications that they could develop that.
Oh, that's very cool. Okay, is there any more questions? Okay.
Yeah, thanks, Mark. It's really interesting. Do you focus only on therapeutic research?
Well, so you cannot only focus on therapeuticals. So our main business is therapeutics. Why? It's quite simple. If you look at the isotope chart and you look at the neutron activated isotopes, they're all therapeutics. So you're basically we have a nuclear reactor here, so our main priority is the therapeutical isotopes. That being said, in parallel with developing a therapeutic, we also try to go on the isotope on the imaging isotope as well. But for us, it's a lot easier. We don't have a cyclotron, so it's a lot easier for us to get these neutron activated isotopes than the proton activated isotopes. And that's in general term. So we focus on it, but it's not our main focus. So it's more in parallel with, we're going to develop a terbium radiopharmaceutical, for example, terbium 161 radiopharmaceutical. We will work with terbium 155, or we will work with other isotopes, indium, or to image, but that's always going to be in service of the therapeutical radionuclide or radiopharmaceutical.
Okay, this leads me to the other question. For the lutetium used, what is the tracer used for diagnosis?
So, and that's a bit the issue now. So they use gallium. So the theranostic pair with lutetium is gallium-68, which works very well. It's a PET isotope, but it only has, if I'm not mistaken, 60-minute half-life, 60-minute half-life. So the problem there is that you can only see what happens in the first hour after injection. So you'll get an idea on the distribution or the initial distribution and some on the accumulation. But if you really want to do dosimetry based on that gallium, you have far too little time points. So yeah, for now it's good for patient selection, but that shift might have to be made for lutetium to go for a little longer lived isotope.
Yeah, taking this disadvantage of gallium that you need to take an image within 3 hours. Do you think zirconium-89 would be a surrogate to gallium-68?
Yes, there's a lot of, the only difference there, or the disadvantage of zirconium is really emerging a lot. The only difference there, it has the chelators do not match. So the zirconium is a bit more difficult to chelate. So what you have then is that you don't have chemically identical compounds. So now you can basically use dotatate. You can just slap gallium on it. You can slap lutetium, but chemically speaking, they're almost the same. They're both 3+. They're both, so chemically your body will not make difference. But with the zirconium, you have to use an entire different chelator because the conventional chelators do not work. So that's a disadvantage of zirconium, but it is boosting a lot. So the zirconium is growing and there's a lot of interest in that. And I think it certainly has its merits, but it also has its disadvantages. So zirconium has a longer half-life, but yeah, with a different chelator chemistry, it does not have to, but it could in theory affect your biological effect of your carrier.
Yeah, so they used to chelate with PFO, peroxamine, and nowadays, recently published, they can use DOTA indeed to chelate zirconium-89 using for prostate. So
yeah,
they can detect the metastasized that can appear after few days, and they can get off the issue with urine because sometimes obstacle to get the entire metastasized.
Yeah, no, I agree. I mean, certainly if you want to do decent dosimetry, that's a lot easier. I mean, if you want to, at some point, the field needs to move towards a thorough dosimetry that you can only do by doing an imaging scan where you can have enough time points so you can basically plot mathematical formulas on it to do some modeling on what is your area under the curve for the tumor and what it is for kidneys, what it is for liver and all these organs, and to try to estimate what the dose is for these patients. And then you can actually look at and calculate, okay, this therapeutical window will work better for you or for you. So that's where we need to, at some point, the field will have to move towards a really even more personalized medicine approach where every patient will get treated differently. But yeah, that's, as I said before, it's very nice to say that in theory, but hospitals also need to pay these kind of scans and they need to pay all these things. So that sometimes is now a bit of a struggle in hospitals itself to not do that.
Thank you. I'm really sorry, but that's all the questions we've got time for, unless it was really quick.
Yeah, I was just going to say, if you're already treating people with lutetium PSMA, why are you not just using the 3 time points on a SPECT scanner from a lutetium to look at your pharmacokinetics?
Yeah, because then it's already too late. So I mean, then you already injected the lutetium. Right? You can use a lot of these data are used for modeling, and for research. That's very interesting. But if you already do your lutetium and you inject that patient, yeah, you can follow up the lutetium, and then you have an idea of the dose it got, but you cannot predict the dose. So it's already a bit too late. But of course, and that's a bit what's might be going on or shifting now already, is by giving that first dose a standard and then go look at the dosimetry and then see, okay, can we now adapt the second dose?
Okay, it's coming up to 2 o'clock, so we'll have to call it there. So Rachel will email everyone with a link to watch the recording if anyone wanted to watch this back. And I just want to say thank you very much for this really interesting talk from Martin. So thank you.
Thank you very much, and for the forum. And yeah, if you have some questions, my email address is on the slide. So feel free to email. I'm always happy to discuss about this.
Amazing, thank you. So I'll pause the recording.
Okay, then yeah, I wish you guys all well, and